So today we're going to start looking at the cell structure and function of eukaryotic cells. If you'll remember, last time we talked about prokaryotic cells, and we looked at those two in the larger picture framework of the three-domain system. So our prokaryotes were in the domain bacteria and the domain archaea. Now we're going to shift over to eukaryotic cells. Easy to remember, they're in the domain eukarya. So let's start looking at the cell level, comparing the three-domain system. So we're going to look at the cell features as they are found in the three different domains. So for our type of cells, we were talking about bacteria and archaea are both prokaryotic cells. And eukarya is eukaryotic cells. Our peptidoglycan walls, if you'll remember, that was found in bacteria, not in archaea, and not in eukarya, which for some reason my little no doesn't pop up till later. I'm not sure why. When we look at membrane-bound organelles, that membrane, remember, is the phosphobilipid membranes, and that is usually found around the nucleus, the mitochondria, chloroplast, that sort of thing. And I don't know if you remember, but that is what separates the eukaryotic cells from the prokaryotic cells. So membrane-bound organelles are not found in bacteria or archaea, but are found in eukarya. And then the ribosome weight. If you'll remember, we talked about all cells have ribosomes, but our bacteria and archaea have smaller ribosomes, whereas eukarya have larger ribosomes. And when we start comparing that, we find that archaea and eukarya are a little more closely related based on their ribosomal RNA than any of the other domains are related. So archaea and eukarya more closely related because of the ribosomal RNA that we find. Introns, we haven't gotten into those too much yet. They're a portion of the DNA that we will get into much later in the semester, but they're not found in bacteria. Sometimes they're found in archaea, and they are present in eukarya. And the different organisms that are found in these domains, our bacteria, clearly enough, are found in the bacteria domain. And in archaea, we have the extremophiles, so those that really liked hot conditions or salty conditions, that sort of thing, are found in archaea. And then eukarya are the things that you are used to. So we're going to find eukaryotic cells in all of the organisms in domain eukarya. So that's our protists, our fungi, our plants, and our animals. So how did eukaryotic cells even come to be? There is a theory on this, that it was produced through endosymbiosis. And if you remember what endosymbiosis is, or if you've never heard it before, that basically means an engulfing of something by the membrane basically folding in and around that object and encasing it in a membrane, and then it comes inside the cell and is wrapped in that membrane. So in the 1970s, a lady named Lynn Margulis came up with what we call the endosymbiotic hypothesis to explain how eukaryotic cells came to be. And so basically what we started with was what we call LUCA. The last universal common ancestor is what that stands for. And it basically means it's a prokaryotic cell from way back when that, we don't know much about. But we think that that's a prokaryotic cell that over time acquired organelles by engulfing other prokaryotic cells that had certain functions. So at some point in time, they would have engulfed a purple aerobic bacteria, because it already had features of our present-day mitochondria. So that first prokaryotic cell would be engulfing the prokaryotic bacteria, and when it does that, it encloses it or uses endosymbiosis to bring it into the cell, wrapping its membrane around it. So some evidence for that is that mitochondria have two membranes. So that would be the original prokaryotic membrane, and then the outer membrane being the Leuca prokaryotic cell having used endosymbiosis to wrap it in that second membrane. Mitochondria also have their own ribosomes and their own DNA, and they can replicate by themselves like an individual cell can do. So it's thought that that occurred, and then later the eukaryotic cell that already had a mitochondria then engulfed a cyanobacteria, which we talked about before. The cyanobacteria can photosynthesize, and so those functions then became the chloroplast. And again, that means that it has two membranes, like a chloroplast does. It has its own ribosomes, its own DNA, and can replicate by itself. So all that evidence led Lynn Margulis to come up with the endosymbiotic hypothesis. So I'd like you to take a moment to look at this YouTube video. It is a good illustration of the endosymbiotic theory, and then we'll get back together and talk about it. So either click on this link or cut and paste it into another browser page. But go take a look at that and then come back here and we'll talk some more. Okay, now that you've had a little bit of time to look over the supporting evidence for the theory of endosymbiosis, I want you to take a moment just to think about reasons that you can think of why that theory might be incorrect. Because something that is important to me in these lectures and in our classroom is that you think critically. So I want you to be thinking about, just based on your knowledge, what reasons could you come up with for the theory of endosymbiosis to be incorrect. And what I'm going to have you do with that is I would like you to write just a short paragraph as to why you think it might not be correct, and email that to me. And I will find a way to put it into your grades as well, whether that be extra credit or as an additional assignment. But I want you to just think critically for a moment. This is a theory that was proposed, I'm pretty sure, in my lifetime. And so, you know, it's a still relatively new theory. Hey, don't laugh, I'm not that old. And so I want you to be thinking about just things that you know that maybe don't make sense to you in this theory, reasons that you might think it's incorrect. And there's no right or wrong answer here. This is just, I want you to take some time to think critically about it. Okay, I can't wait to see what you guys say about that. So looking on this particular chart from left to right is time. So we start with the first cells basically in this far left, and these would be what we call Leuca, remember when we first talked about that? That would be the last universal common ancestor. And then, of course, the breakout between archaea and bacteria. And if you look sort of in the center section, this is where eukarya is going to branch off. And so our eukaryotic cells flowing this way, this is where our aerobic bacteria was engulfed and thought to be the mitochondria. And then later we would have the engulfment of the chloroplasts, or the cyanobacteria that produces a chloroplast. So that leads to our autotrophic eukaryotic cells. We're going to start going over the features specifically of eukaryotic cells. And just to start with, I just want to point out that note that every eukaryotic cell is encased with a phospholipid bilayer membrane. And we're going to actually spend an entire lecture on membrane structure and function, so I'm not going to get into detail about that today, but those membranes are super important. And the biggest function for those in the eukaryotic cell is that there are membranes that enclose each organelle. So eukaryotic cells are very compartmentalized, which allows each of those organelles to perform its function and to keep all of the proteins that they need close by. And so that compartmentalization makes this cell more efficient. And there are ways that proteins and other biomolecules can be transported between the various organelles, and we'll talk a little bit about that. So today we're going to talk about the general features of eukaryotic cells. Now, what I have on this particular slide is an animal cell, and animal cells and plant cells are both eukaryotic cells. So as we go through the different features, I will point out some of the differences between the plant and animal cells, and at the end probably do a summary so that we can compare and contrast those. So let's start with one of the largest organelles within a eukaryotic cell, and that's the nucleus. The exception to that would be in plant cells we tend to have extremely large vacuoles that are larger than the nucleus. But for the most part in animal cells, the nucleus is going to be the largest organelle that you see. It's usually this center, it's usually in the center of the cell. So here it is on our image here. And the nucleus is sort of like command central for the entire cell. It contains most of the genetic information of the cell. It controls gene expression and therefore metabolism in the cell. It mediates the replication of DNA during the cell cycle, so it allows for cells to divide and to get the same amount of genetic material as their parent, except for sperm and egg cells do get half of the DNA, and that's very controlled, and it's controlled by the nucleus. The nucleus also produces mRNA, which is messenger RNA, and then it contains the nucleolus. So the nucleolus is this center part right here, and it has its own function. All right, let's take a look at this nucleus in detail. So, like we said, it's the largest organelle, and all organelles are membrane bound. And if we put this nucleus in the center of a cell, that means that it's surrounded by the cell's cytoplasm. And the cytoplasm is just that liquid portion of the cell. It's made up mostly of water. Within the nucleus, we have something similar to the cytoplasm called the nucleoplasm. And the nucleoplasm, again, is mostly water-based, and it's just the liquid portion of the nucleus. So in order to separate these two and to compartmentalize the nucleus, we have what we call the nuclear envelope, also called the nuclear membrane. And so this inset over here shows us a blown up image of the nuclear membrane. And it is composed of a phospholipid bilayer. So we have the hydrophilic heads on the outside and inside. And that's because they interact well with the cytoplasm and the nucleoplasm, which are mostly composed of water. Then we have our hydrophobic tails that point towards the center. So that makes for a two-layer membrane, separating the nucleoplasm from the cytoplasm. So let's talk a little bit about what's in the nucleus. The first thing we're going to notice is chromatin. There's lots of chromatin all throughout the nucleoplasm. And what chromatin is a combination of proteins and nucleic acids. And just prior to the cell dividing, these chromatin condenses into chromosomes. So that might be a term you're a little more familiar with. If you've ever talked about Down syndrome, you know that there's a portion of a chromosome that's missing in that. So it's the genetic material. Those chromosomes are made up of genes, which are the basic units of heredity. And so that's what we have within the nucleus, our genetic material. Now, that genetic material, when we need to start using it, what we call translating it, into proteins, into signals that tell the rest of the cell to produce proteins, to change their metabolism. In order to do that, the nucleus needs to recruit certain proteins from the cytoplasm. And so if you look at our image here, we have what's called nuclear pores within the nuclear envelope. And we can also see this in our inset. Now, what I don't want you to walk away with is thinking that the nuclear pores are just holes in the membrane. There are no holes in the membrane. There are what we call transport proteins that cross both layers of the membrane. And they regulate what comes in and out of the nucleus. So those proteins that are produced in the cytoplasm that need to come into the nucleus usually have some signal on them, or they might have a particular shape that allows the transport proteins to say, okay, you can come into the nucleus. The other part is that the nucleus produces messenger RNA. And messenger RNA is essentially what is required to, excuse me, to produce a sequence of amino acids to make a protein. And all that occurs outside of the nucleus. So mRNA that is produced within the nucleus needs to be able to cross these nuclear pores as well. And they often are accompanied by what we call a signal protein that tells this transport protein it is okay for the mRNA to exit the nucleus. So it's a very regulated transport of items inside and outside of the nucleus. Now, what you might also notice is right here a very densely packed chromatin area, and it's called the nucleolus. And it is not separated by a protein, it's just a portion of the nucleus that has densely packed RNA. And it's a particular type of RNA that's produced right here. It's called rRNA or ribosomal RNA. So ribosomal RNA plus certain proteins form the subunits of ribosomes. So again, that rRNA is going to need to exit through one of these pores as well. Now, interestingly enough, and I'll show this in a couple slides, but the nuclear envelope is actually continuous with what we call the endoplasmic reticulum. So if I were to draw it, it might look a little bit like this, and you will soon learn that I am not the best artist with these particular pens. But the endoplasmic reticulum membrane is continuous or contiguous with the nuclear membrane. So when we have subunits of ribosomes coming out, they actually are going to be a part of the endoplasmic reticulum, and we'll look a little bit about that in just a second. But just so you know that those two membranes are contiguous. So before we move on to ribosomes, I just wanted to give you an idea of what the nucleus actually looks like in a real cell. So this image on the left is a nerve cell from an animal, and you'll see the nucleus in the very center of that cell. And these nerve cells are actually quite large here, so they make it easy to see. Also on the right is an onion cell, and you can see the nucleus there as well. And so they are a large portion, probably very large organelles within these cells. What you can't probably see too well right here is that in an onion cell, there's a large vacuole that probably takes up this entire portion right here. So it's not the largest organelle in a plant cell, but it typically is in an animal cell. Because we just talked about the nucleus, I want to talk about the ribosomes. And that's because we talked about the ribosomal subunits being assembled in the nucleolus, or at least the subunits of them. So ribosomes are the site of protein synthesis. So every protein that's made in your body is made within these ribosomes. And they are composed of a large and small subunit, and they are often associated with what we call the rough endoplasmic reticulum. And the reason it's called rough is because it contains ribosomes. So those are right here, and remember we talked about this endoplasmic reticulum. The membrane is contiguous with the nuclear envelope. And so most of the ribosomes are going to be associated with the rough ER. They can occur individually within the cytoplasm. Oftentimes they associate together, so just maybe a string of ribosomes, and we call that a polyribosome. So they can be found there as well. So here is our ribosome with its two subunits. It has a small subunit down here on the bottom and the large subunit on the top. And this ribosome is currently in the cytoplasm. So in this image, I've pretty much said anything below the blue line occurs in the cytoplasm. Anything above the blue line is going to occur within the endoplasmic reticulum. So let's start at the beginning of the process. So messenger RNA from the nucleus is transported through the nuclear pores into the cytoplasm. And on that mRNA is all the information that the ribosome needs to create a growing protein. So in the cytoplasm, the ribosome attaches to the mRNA and it begins the process. But that process is halted temporarily by another protein, and that's called the signal recognition particle. And that signal recognition particle binds the peptide that the ribosome was beginning to create. And when it does, it delivers it to a receptor protein on the endoplasmic reticulum. And it attaches, so it allows the ribosomes to be attached to the membrane of the ER, the endoplasmic reticulum. Okay, once it does that, that signal recognition particle then comes off of the polypeptide, and we continue the process of making a protein. So if you look at our diagram, we have the mRNA down on the bottom, and you'll notice that it has all the nucleic acids that you are now familiar with and you know the structure of. And then what happens is on the mRNA, a transfer RNA binds the mRNA. So the way DNA works is every 3 nucleic acids signal an amino acid. So this transfer RNA takes the signal of these 3 UCA nucleic acids and says, okay, that goes with this particular amino acid. So it will bind the mRNA but bring in the amino acid. And as it does, the amino acids begin to bond chemically to one another. And as we said, amino acids all in a line start to form our protein. So here is that primary structure of a protein. So if you think back to when we were talking about proteins, they have primary structure, just means the direct amino acid chemical bonds that are signaled for on the messenger RNA. So once we have that growing strand of primary protein, as that's happening, all of this, once we get to the end, gets cleaved off, and it's within the inside or the lumen of the ER. Within the ER is where the protein folding occurs. Remember, we talked about secondary, tertiary, and quaternary structures. This is where that happens. So within the lumen of the ER, all that folding is going to take place, and we're going to have those chaperone proteins that come in and say, oh wait, that's not done right. Let's make sure it's done correctly. So that's how proteins are synthesized on ribosomes. Next, we have the endoplasmic reticulum. And the endoplasmic reticulum, as we've said before, is contiguous with the nuclear envelope. So they are located right here. And as you can see, there are 2 types of endoplasmic reticulum, or ER. There's the rough endoplasmic reticulum, and it's just considered rough because it is studded with all those ribosomes that we just talked about. And then there's the smooth ER, which is right here, and they each have unique functions. So here's our image of our rough and smooth ER, and so you notice that all of it is contiguous with the nuclear membrane. And so the rough ER, which we also see on this image to the right, this is what we call an electron micrograph. So an electron microscope was used to look at the rough ER, and you can see all of those black dots are the ribosomes that are on the membrane. So the rough ER contains ribosomes. It is imperative for folding proteins like we talked about. And it also adds chemical groups to proteins. That's where we get certain special functional groups. And then the finished product is usually transported out of the cell, and we will talk about how that happens. But some of the finished product goes on to the Golgi apparatus for more preparation. The smooth ER is a little different. It synthesizes lipids, such as our steroids, for instance, and it assists in transport of macromolecules, again, usually by transporting them through the Golgi apparatus. They also assemble and repair cell membranes. So the smooth ER is just as important as the rough ER, but even though they have a continuous membrane system, they serve different functions within the cell. Next, we're going to look at the Golgi apparatus, and so that is right down here. And I like to call this the mail carrier with special powers of the cell, because it tends to process packages, adjust them a little bit, and then send them to particular places within the cell. So when we're looking at the Golgi apparatus, it looks kind of like a stack of pancakes. And we talk about this stack in its relation to the ER. So the side that faces the ER, this right here, we call that the CIS, C-I-S, the CIS face. The portion of the stack that is away from the ER, right here, this is called the trans face. And that's going to be easy to remember, at least for the trans portion, based on its function. So if we look at proteins that are coming from the endoplasmic reticulum, as well as lipids that are coming from the smooth endoplasmic reticulum, they, at the ends of their stacked membranes, are these vesicles that basically just transport those lipids and proteins to the Golgi apparatus. And note here that the membrane fuses with the membrane of the Golgi apparatus to allow the contents to be dumped into the Golgi. So once that occurs within the Golgi apparatus, the Golgi can modify those lipids and proteins, attach groups to them, attach what we call kind of like address labels for where they're going to go within the cell. And then once they do that, then a lot of times at the ends of those pancake stacks, you'll see more vesicles. So once they've been modified, then they're placed into vesicles to be transported either to a place within the cell or out to the plasma membrane. And when they get to the plasma membrane, the vesicle membranes fuse with the plasma membrane and allow this opening for the contents to be excreted. And that's a process that we call exocytosis. So if you think back to here, when the vesicle merged with the Golgi apparatus membrane, that's called endocytosis because it's dumping those contents into the lumen, whereas the exocytosis, we're dumping those contents outside of the plasma membrane. So if you think about if we had produced steroids within the smooth ER and they get adjusted and tagged, then they can be excreted outside of the cell membrane and transported to other parts of the body. Now the other part of the Golgi is that they also produce their own type of vesicles that aren't related to the products from the ER, and we call those lysosomes, and they have a very specific function. The lysosomes that are specialized vesicles produced by the Golgi contain what we call hydrolytic enzymes. They are filled with an extremely low pH fluid where these hydrolytic enzymes can live. And the function of these enzymes is to digest or break down materials. It's what we call autophagy. So if there are portions of the cell that have been damaged or proteins that have been folded incorrectly, they are engulfed in these lysosomes and broken down and destroyed, and then their parts can be reused. So oftentimes our cells will detect bacteria or viruses that are dangerous to the rest of the system, and they will be signaled to bring them in and send them into lysosomes. And that's particularly true of white blood cells that are important in destroying foreign objects. So white blood cells, for instance, have a larger number of lysosomes than most other cells. But those lysosomes are created in the Golgi apparatus. Peroxisomes are another type of vesicle that we find within a cell, and they are membrane-bound organelles. And so, unlike our lysosomes, which are produced by the Golgi apparatus, peroxisomes are actually produced by free ribosomes. And they also have a breaking down feature. So they oxidize fatty acids, and in doing so, that produces hydrogen peroxide. That's where we get the name peroxisomes. But they also contain enzymes that then further break down the peroxide into water and oxygen. So we take fatty acids, which can be built up a little too high, and break them down through a process that brings them to water and oxygen, which are perfectly fine and useful for the cell. Our next organelle that we're going to take a look at is the vacuole. And note that I have switched over here to an image of a plant cell, because there are some animal cells that have small vacuoles, but they're fairly rare. Our vacuole are found mostly in plant cells. And not only are they just found in plant cells, they take up about 90% of the volume in our plant cells. So here in our image, just to kind of get you oriented, we have the nucleus right here with its endoplasmic reticulum near it, but this large portion right here is the vacuole. And again, it has the bilipid membrane separating it from the rest of the cytoplasm, and it has several functions. It's mostly a storage function. It stores water and sugars, salts. It sometimes can store pigments. For instance, if you are looking at a pink flower, those pigments that make it pink are usually found within the central vacuole of a plant cell. It can also contain toxins. So if you have ever rubbed up against a plant that made you itch, or that would make you sick if you ate it, those toxins are normally held within this vacuole. But the big role of the central vacuole in a plant cell is for what we call turgor pressure. So if you can imagine, if we took the water out of this vacuole, all the contents of that cell would kind of implode. And so the whole of a plant gets its turgor pressure, or its rigidity, from keeping water in that vacuole that holds the cytoplasm and pushes it out towards the cell walls. So it allows the plant structure to be more rigid. So that's especially important for non-woody plants. Once we have wood within a plant, then that's a different type of structural support. But many of like your flowering plants that you would, if somebody buys you flowers, that's not going to be a woody plant. They're not bringing you a tree, for instance. And so in order for it to remain upright, it requires this turgor pressure within the vacuole of each cell to allow for that turgor pressure or rigidity of the plant. So while we're looking at things that are unique to plant cells, I did want to mention the plastids. And this is just a group of organelles that are membrane bound within plant cells. The most well-known of those is the chloroplast. And within the chloroplast that you can see here under letter L, the chloroplast uses pigments to capture solar energy and use that to synthesize carbohydrates. And they do that by taking carbon dioxide out of the air, and they convert it to sugars. And we're going to go into photosynthesis in detail a few lectures from now, but just for now to know, chloroplasts are some of the best known plastids in plants. But there are other plastids within plant cells, and I'll just briefly touch on those. But for now, let's get to know the chloroplast better. So let's take a look at the chloroplast and at the anatomy of a chloroplast. Just like all the other organelles that we've been talking about, it has a phospholipid bilayer, so that is right here. And then there are some other membranes within the chloroplast, and they're arranged in these discs that we see right here. Each one of these discs is called a thylakoid, and it's composed of a membrane and a lumen. A lumen is the inner space within the membrane. And the thylakoids are where the chlorophyll A, chlorophyll B, and various accessory pigments are going to be found. That's where the chloroplast can collect the solar energy from the sun. And when we stack up these thylakoids together, we call that a granum. And as you can see, there are multiple granums throughout the chloroplast. The other part of the chloroplast that we want to mention is the stroma. And the stroma is that liquid substance, sort of like the cytoplasm of the cell. And remember, in the nucleus, we called that nucleoplasm. We made that nice and easy. Well, what it's called in the chloroplast is the stroma. And the stroma is where all the proteins are going to be located for carbohydrate synthesis. So we collect the energy in the thylakoids, and then that's used to convert carbon dioxide and water into our carbohydrates. So that's where that takes place. So if you look over here, this is just an electron micrograph. You can see these slight stacks, the granums of our thylakoid, and those are actually connected as well. So they are all connected by these tubes. You can see them here. So that is the anatomy of our chloroplast. Other plastids in plants are called amyloplasts. Amylo meaning starch, and plast for our plastids. And so in this image to the left, this is a potato that's been thinly sliced and then stained with iodine. Iodine, when it interacts with starch, turns a dark purple color. So you can see the plastids within these cells. If you look, I'll show you the outline. So this right here would be the outline of this particular potato cell. And each of these are amyloplasts that contain starch. So in a potato, they're used just to store that extra food. The image on the right is a picture of a root tip. And so there are highly specialized cells at the bottom of a root, so right here, and they're called columella cells, and the reason they're called that is because they're columnar shaped, so like that, for instance. And they contain amyloplasts as well, which have that heavy starch grain, but they are used specifically to sense which way is down. So we call those gravity-sensing amyloplasts. And so if we were to turn the root tip on its side, those heavy starch grains in the amyloplast would fall. And there are sensors within the cell membranes that allow that root tip then to know that it's changed directions and that it needs to start growing towards gravity. All right, next is our mitochondria, and note that I've gone back to animal cells. Mitochondria are present in animal and plant cells both, so don't get confused by that. But here they are. There are pictures of them here and here. The shape of them is kind of like a capsule, or like if you needed to take a Tylenol, about the same shape. And they are what we call the powerhouse of the cell because they produce ATP. And if you'll remember when we talked about that in our biomolecules lecture, that the ATP has 2 phosphate bonds that are extremely volatile and easy to let go of energy because they have that high energy bond. And so the mitochondria is the site of what we call cellular respiration, and we're going to go into a lot of detail about that in another lecture as well. So right now let's just look at the anatomy of mitochondria. So when we look at the anatomy of the mitochondria, just like every organelle, it has 2 membranes, that phospholipid bilayer membrane. So it has the outer membrane, and then its inner membrane is a little bit unique. So the inner membrane has all these folds to them. If we're talking about one fold, we call that a crista. If we are talking about a group of them or more than one of them, we call that a cristae. So those are multiples, we call that cristae. And those inner membrane folds the cristae are actually very important. They are the site of electron transport chain system, and when we start to talk about cellular respiration, you'll realize how important that is. But those folds, those cristae, increase the surface area. So the more we have, the more output we can get from our mitochondria. So that's why that's important. So the space on the inside of both of those membranes is called the matrix. Okay, so no Keanu Reeves here, but the sort of similar to the cytoplasm, this is the fluid within the mitochondria. And so there are some ribosomes in there. There are also what we call ATPase, which is a protein that is involved in synthesizing ATP. And remember also that the mitochondria has its own DNA, so you will see that in there as well. The cytoskeleton is important for all eukaryotic cells. Just like we have a skeleton that helps to support our body, so does the cell on an individual level. And there are three major components to the cytoskeleton. Those are the actin filaments, the microtubules, and the intermediate filaments. So the actin filaments, which on the right here are stained in green, these are a bunch of cells. You can see the blue is the nucleus. So the green is your actin filaments, and they're the smallest of the three parts. And they usually form a network that lays just under the plasma membrane, so it helps to keep that structure in place. Now, depending on the cell type, actin is very important in like muscle movement, for instance, actin and myosin together, moving things along. That's probably not something we're going to get into in our particular class, but just so you know, these cytoskeletal proteins are really important. And then the intermediate filaments, these are slightly thicker, and they're really named because they are in between the sizes of the actin and the microtubules. And again, they provide a network of support. And then for our microtubules, these are even larger. And microtubules, again, provide support. They tend to be close to the nucleus. They usually come together in what we call the microtubule organizing center. And the main microtubule organizing center is what we call the centrosome, and that's the one that stays right near the nucleus. Now, the nice thing about microtubules is they are made up of proteins called tubulin, so that makes sense, right? And there are certain motor proteins that work on microtubules, and so microtubules essentially form tracks throughout the cell that the organelles can be moved upon using those motor proteins. So within a cell, things are constantly moving. Everything is dynamic, and nothing really stays in place. So what we call this is cytoplasmic streaming. So we talked about how oftentimes microtubules are used as tracks for the organelles to move on. Actin and microtubules both tend to facilitate this cytoplasmic streaming, this movement throughout the cell. So I'd like you to just take a moment to look at this YouTube video, because it's very hard to describe unless you actually see a video of it happening. Like, the best thing that I can compare it to is how blood moves throughout a person's body. It's very similar on the cellular level. So as we just look at plant cells versus animal cells, both of them are still eukaryotic cells. So if we look in the center of this Venn diagram, this is what both cell types have in common. They all have DNA, have a nucleus, have a nucleolus for that matter as well, a cell membrane, the mitochondria, cytoskeleton, there it is, rough ER, smooth ER, cytoplasm, ribosomes, and a Golgi apparatus. What is unique to plants is the cell wall, which we really haven't even talked about, but it's here on the right. It's the very outermost layer, so it's outside of the cell membrane, and it is composed of cellulose, which is a much more rigid structure, so it helps to protect that cell. Obviously, plant cells have chloroplasts, as well as other plastids, and a large central vacuole. On animal cells, what they have that plant cells do not, I'm looking at these and we only really touched on one of them. So the top and bottom, cilia and flagella, those are two modes of transport, basically, two ways that cells move. And so that is found primarily in animal cells. I will say the exception to that is that some plant gametes do also have a flagella, but oftentimes not. The lysosomes are only found in animal cells. And then centrioles, which we didn't actually talk about, but we could have within the cytoskeleton. The centrioles are basically short cylinders of microtubule triplets. So there's like 9 sets of those triplets. And so we see those laying at right angles to one another, and they are a major microtubule organizing center for the cell. We don't actually have those in plant cells. So those are the differences there. So everything that we've talked about today is like for a generalized cell. So if we have an animal cell that's like a cardiac cell, it may have, like, say, more mitochondria than fat storage cells. So there is some fluctuation in the different cell types as to how many of each of these organelles and things that are present. And like I said, some of them even lack certain organelles here and there, but for the most part, everything we've talked about today, we see in eukaryotic cells. So is anybody's mind blown yet as to how complex just one single cell can be? And then if we think of every living thing is made up of these building blocks of cells, it is so complex and so amazing. This slide and the next are just for you to practice if you can figure out what the different structures are in the plant cell and an animal cell. So these are for your practice. Just a hint, you will probably see these again.